to 40 publications. She have 27 popular article. She have wrote so many manuals, book chapters, and she is a major advisor of one student and and the member of other advisory committee of 14 students. She is the recipient of several awards, including Merit Scholarship, Best Paper Award, Best Oral Presentation, Best Poster Award, Women Scientist Award 2017, Inspiring Lady Veterinarian Award, and Young Women Scientist Award. So we have Dr. Savita Kumari, and she is going to present the microbial contamination in milk. So welcome, ma'am. Now you can present your presentations.
Thank you, ma'am. Thank you for giving me opportunity to part of this training program. So here I am with my presentation, and the topic is microbial contamination in the milk. And I will present my presentation with the following heading of the introduction, significance of the microorganism, sources of the contamination, and what are the different types of the microbial contaminants, and which test we can use for the detection of the microbial contamination, and the control measures to curtail the contaminations. So coming to the introduction, dairy is an essential agriculture activity, and it plays a vital role in the global food production system. It plays a significant role in the economy of the India as well as in the globe. And presently, milk production is a major economic activity, and the milk production has increased about 20% in the last decade. And almost 150 million households are engaged in the milk production, and more than 6 billion consumers are there for the milk and the milk products. And among the milk, the bovine milk is most consumed product, and about 40% of the total milk used is from the bovine. So looking in the larger scale production and the utilization of the milk, the quality and the safety of the raw milk is very essential. So for its consumption as liquid form, as for the production of the quality dairy products. And milk is a very nutritious food product, and it is prone for the bacterial contamination. So the hygienic production of the milk is a very challenging task for the global dairy industry. So it is a nutritious substance, nutritious food, and provide environmental conditions for the growth of the many type of the microorganisms. And milk can be contaminated with the biological or chemical origin substances, and the microbial contaminants is one of the major source of the contamination. So why we should study the microorganism in the milk? So if we study the microorganism, it will be helpful for the judging its sanitary quality and the condition of the production. It is milk in the bacteria in the milk is a cause of the spoilage of the product, and sometimes it may causes the certain chemical changes in the milk, which is non-desirable in the raw milk, but it can be utilized for the some beneficial microorganism is also there in the products, which is used for the making of the cheese and the yogurt. So what will be the effect of the microbial contamination? It negatively affects the milk quality and the shelf life. During the refrigeration temperature, some type of the bacteria, which are known as the cyclotolerant bacteria, they can proliferate, and they produce some extracellular enzymes which result in the milk spoilage. And it is also fermentation due to the lactic acid production, and which are having effect on the sensory effect, texture, flavor, and the organoleptic properties of the product. So milk is an important source of the foodborne pathogen also, and it can adversely affect the human health and can cause the various type of the diseases in the human being, like diarrhea, stomach cramping, vomition, and so many other also affect the safety and the quality of the dairy products from the source, and also responsible for the severe economic losses at the various points of the milk production chain. So if we see, the milk is a various contains a diverse source of the microbial population, and when it is synthesized in the udder, it is a virtually sterile. What are the factors which influence the microbial contamination? So first of all is the health and the hygiene of the cow, and the environment in the which cow is housed and the milked, the process which are used for the cleaning and the sanitization of the milking and the storage equipments, and the temperature and the length of the time for which the storage of the milk is done. All these factors combinedly influence the microbial contamination in the milk. So what will be the sources? As I told, the raw milk which comes from the udder is almost sterile, and it contains less than 1,000 of the bacteria per ml. But it can get contaminated from within the udder, from the exterior of the udder, and from the surface of the milk handling and the storage equipments. Microbial contamination sources may be from the air, milk equipments, feed, soil, gases, and the bedding materials. And from milk, various type of the microorganism can be transmitted to the human being through its consumption. So from the interior, from the cow udder, teat surface, it is having a diverse bacterial population, and it can contain the epithelium adhering commensal like bacteria like streptococci, staphylococci, and the micrococci. Sometimes it also contains the pathogenic bacteria which are a source of the infection to the human being, like Mycobacteria brucella. And some other type of the environmental contaminants like fecal E. coli or Campylobacter, Salmonella can also get entry into the raw milk. Sometimes due to the mastitis, important bacteria like Streptococcus, Agalexia, Staphylococcus aureus, and Escherichia coli, they can also get entry into the raw milk. So mastitis is a very important cause of the microbial contamination in the milk, and it is having a very important economic impact on the milk production, as it is reduces the milk yield and affects the dairy processing and the food safety. Most of the common bacterial pathogen associated with the bovine mastitis is the Staphylococcus aureus and the Streptococcus agalexia, and also the E. coli. Sometimes environmental pathogens like E. coli and the Streptococcus uberis, they can contaminate the udder and cause mastitis also. And it is in various studies, it is found that the subclinical mastitis, where obviously the clinical sign of the mastitis is not visible, they are responsible for the 3 to 5% of the total milk loss yield, and where the somatic cell count is about the 2 lakh cells per ml. So what will be the effect? If udder, even the udder is contaminated with the bacteria, and it causes the change in the milk yield and the composition. So the milk yield will be low, somatic cell count will be high. It will also affect the milk composition. It will affect the serum protein and will release some proteolytic enzymes. Sometimes bacteria, they release the toxins and the abnormal protein, which may influence the milk flavor and the taste as well as. So this all about the contamination from the interior of the udder. Now comes to the exterior of the udder. On the exterior of the udder, from where it comes, bacteria comes naturally from the environment. And when the teat gets soiled with the manure, mud, feeds, or the bedding material, it affects the teat and the udder of the uh, cow. And both type of the bacteria, like thermoduric and the psychotropic bacteria, they are common on the teat surface. So water and bedding material which is used in the milk production, it should be of the high microbiological quality, because it can be a source of the pathogenic as well as the saprophytic bacteria. And several studies have highlighted that the water used in the farm has a major role for the pseudomonas raw milk contamination. Bedding material, especially during the winter season, it can carry the high bacterial load and can contaminate the teeth. Coming to the equipment, cleaning and the sanitizing procedures, they can also have a role in the microbial contamination. And what happens when the equipment is not cleaned properly, the milk residues which are present on the contact surface, they support the growth of the variety of the microorganism. And the soil surface of the equipments, they are contaminated with the bacteria. Water which is used, that can also source of the bacteria. So the effective use of the chlorine or iodine sanitizers are used or recommended to reduce the level of the bacteria, especially the psychotropic bacteria. Coming to the milk storage temperature and the time, refrigeration temperature, it is having an effect on the growth of the bacteria. It prevents the growth of the non-psychotropic bacteria, whereas the psychotropic bacteria, which can grow on the refrigeration temperature, they can flare up at the refrigeration temperature. And but these are not the thermoduric, means they can be destroyed by the heat treatment. When the pasteurization is done, these organisms usually they can destroy. So what are the different types of the microbes in the milk? In milk, diverse microbiota is present. Various type of the viruses, mold, yeast, and especially bacteria, they can contaminate the milk. And many among the bacteria, gram-positive bacteria, gram-negative bacteria, they are present, and their content, their ratio can change on the basis of the temperature or the storage. Whereas as in the fresh raw milk, gram-positive bacteria, they predominant, where in the cold storage, gram-negative bacteria, they increase in the number. So coming to the viral organism, not only the bacteria, milk can also get contaminated with some of the viruses, important viruses. Some of these viruses, they can also cause disease in the human beings. And when studies is done, it is found that the 20 to 35% of the milk samples from the intramammary infections or the high somatic cells, they are negative for the bacteriological sample analysis. So that it means that they can either contain the mold or yeast, or they can contain the viral etiological agents also in the milk. Some of the viruses which can be present in the milk are the hepatitis C virus, FMD virus, cowpox virus, tick-borne encephalitis virus, and the Rift Valley fever virus. So what are the different types of the bacteria present in the milk? Among all these bacteria, one of the important bacteria is the psychotropic bacteria. Pseudomonas and Acinetobacter, these are the example for the psychotropic bacteria. And these psychotropic bacteria, they can grow and the proliferate in the low temperature and the spoil milk by the producing extracellular lipase and the proteases. In the dairy industry, Generally, milk is stored at the refrigeration temperature below the 10 degrees centigrade before the processing for the 2 to 5 days. And what happens? This is for the slowing down the production of the or growth of the pathogenic bacteria or the thermophilic bacteria. But this also creates a conducive condition for the development of the psychrophilic bacteria or the psychotropic bacteria, which can continue to grow at the low temperature, up to the below the temperature of the 7 degree, and can eventually dominate the microflora. So the most psychotropic bacteria found in the milk, they can produce the hydrolytic thermostable enzymes which break down the milk major constituents like the milk fat, milk fat, proteins, and the lecithin. And generally, these hydrolytic enzymes like the proteases and the lipase, they withstand the thermal treatment and are responsible for the off flavors, bitterness, and the textural changes in the dairy products which are totally undesirable. Sometimes in the milk, lactobacillus, they can also cause the milk spoilage at the room temperature. And at the temperature of the 10 to 37 degrees centigrade, souring of the milk occurs mainly due to the Streptococcus lactis, Enterococci, lactobacilli, and the other coliform of bacteria. So the other bacteria are thermoduric bacteria. Thermoduric bacteria means they resist the pasteurization process of the milk and use the components such as the protein and the fat as a nutrient source, and causes the organoleptic changes and the reduced product shelf life. One of the example of the thermoduric bacteria is the Bacillus cereus, and other type of the Bacillus and are also responsible for are as the thermoduric organism. And not only the bacteria, their spores can also these are the spore forming bacteria, and their spores can also be responsible for the contamination of the milk. And these bacteria, these thermoduric bacteria, from where they arise, these are commonly present in the soil, bedding material, feed, dust, and which all they can contaminate the cow teat, and from there they can contaminate the raw milk. So the pathogenic microorganism in the milk, number of pathogenic microorganisms are present in the milk, and they can cause the foodborne illness due to the consumption of the raw milk or the dairy products which is made with the such type of the milk which is not properly pasteurized, or due to the post processing contamination. Poorly, when they are handled poorly after the post processing, they can also get contaminated with the pathogenic microorganism in the milk. And and from where they come, like the water, food, and the environmental factors like the coliform, E. coli, and other enteric bacteria, they can introduce into the milk. So these are of the major concern, and because they are responsible for the foodborne illness and affects the human health. And approximately 90% of the dairy related or the diseases which occurs in the human are from the pathogenic bacteria in the milk. So we can imagine the significance of the pathogenic bacteria in the milk. So what are the major pathogenic bacteria, foodborne pathogens, which are associated with the milk, like Campylobacter, and these causes various type of the diseases like the gastroenteritis. Some of the bacteria like Coxiella burnetii, they cause the Q fever. Some E. coli, they cause hemolytic uremic syndrome. Mycobacterium bovis or Mycobacterium tuberculosis, which are responsible for the tuberculosis. Mycobacterium paratuberculosis, which is linked to the Crohn disease in the human beings. Salmonella, which is responsible for the gastroenteritis or the typhoid fever. Yersinia, they are responsible for gastroenteritis. Brucella, one of the important milk pathogen, which is responsible for the fever, joint pain, and the fatigue in the human beings. Listeria, which is responsible for listeriosis, and associated with the flu-like symptoms and the abortion in the human beings. Apart from the bacteria, milk can also get contaminated with the molds and the yeast, and mastitis, fungal mastitis, is one of the major source for the contamination of the molds and the yeast. Approx 2 to 13% of the cases of the total mastitis in the cow is associated with the fungal infections, and among the fungus, Aspergillus, Fusarium, Penicillium, they can grow in the milk, and not only they can grow in the milk and produce the detrimental effects, but they can also produce the mycotoxins, which are of a major concern of the health, and it produces a health hazard. Apart from the molds, some yeast are also responsible for the contamination, like Candida and the Cryptococcus neoformans. So, how the microbial contamination in the milk can be tested? There are some tests, like microbial tests, like direct microscopic count, standard plate count, dye reduction methods, coliform test, yeast and mold count, and the detection of the pathogens in the milk. So what is the direct microscopic count? When direct microscopic count means it enables the direct rapid enumeration of the bacterial cell. How it is done? A fixed amount of the volume of the milk, say 1 microliter, is spread over a area, and it is dried and it is stirred, and then it is seen under the microscope for the somatic cells count and the shape and the arrangement of the bacterial cell present in the field. And with the some formula, total microscopic count is calculated. Another method is the standard plate count, SPC method. And in this method, milk is serially diluted, and each dilution is poured on the petri dish, and then agar is poured and mixed and allowed overnight for the growth of the bacteria. And the bacteria grown is measured in the coliformic unit. And based on the number of the bacteria, grading of the milk is done. And if the milk SPC count is less than 2 lakhs of the milk, then the grading of the milk is very good. If it is 2 lakhs to 10 lakhs, then good. 10 to 50 lakhs, then it is fair, and more than of the 50 lakhs SPC count is the milk is considered as a poor. Similarly, such type of the plate pouring methods can be used for the coliform count and the yeast and mold count also. But the media is different and specific according to the bacteria or the yeast or mold to be identified. So standard plate count, it represents the bacteria which can grow between the 30 to 35 degrees centigrade under the aerobic conditions, and they reflect not only the hygienic condition of the milk, but the way how the product was handled. Because the higher value of the species in the milk indicates the poor handling practices during the milk production, and it can also be a risk for the presence of a pathogenic microorganism in the milk. So it reflects the efficiency of the cleaning procedure and also the storage temperature and the hygienic conditions of the udder during the milking. Coliform count, it estimates the number of the coliform bacteria in the milk, and coliform mainly originate from the cow environment, and it is indicator of the fecal contamination, usually from the soiled udders and the teats. And high level of the coliform count in the milk can indicate the poor environmental hygiene, poor hygienic milking practices, and the further handling. And improperly cleaned milk equipments can also be associated with the higher number of the coliform count. Contaminated water, inadequate refrigeration, and the presence of the coliform So high public health significance like the Shija toxin production producing the E. coli. Another method is the dye reduction test. One of the methods milk. Normally, what happens? The milk which is produced under the under 10 to the power 5 into 10 to the power 5 CFU per ml the milk. And MB methylene blue reduction test. It is a quick method to assess the microbiological quality of the raw milk and as well as the pasteurized milk. And in this, methylene blue is used, which is a blue color dye. And when it is added to the milk, all the milk contents becomes blue. And when the oxygen present in the milk is due to the microbial color of the dye, it gets color of the dye. It is for poor quality milk. When 1 to 2 hours, then the fair quality of the milk. 3 to 4 hours, if it takes, then the quality of the milk is good. And if the uh, dye reduction takes 5 hours or the above, then the quality of milk is very good. Another dye reduction test is the rizaurine reduction test, and it is a in this various discs are present in the rizaurine or with the help of the rizaurine comparator and standard rizaurine disc. Color of the reduction in the milk is done. In this test, the reduction of the dye takes place in the 2 stages. First, in the dye is converted to the rizaurine. And then again it converts rizaurine. So the various color which is indicated with the disc color is blue and the disc number is 6, then the grading of the milk will be excellent. In the light blue color is the disc is with the 5 number of discs. Purple pink color and the plus is there, and grading of the milk accordingly done. So how the detection of the pathogenic bacteria is done? Basically, it is done on based on the bacterial culture on the selective agar media. Like if we have to grow E. coli, then we use the selective media, which is eosin methylene blue. Similarly, for staph, mannitol salt agar, on other specific media for salmonella also. And so the traditional method of the identification of the pathogenic bacteria is the based on the isolation of the growth of the microorganism on the solid media, followed by their observation of the morphological identification as well as the biochemical characteristics. And now these polymerase chain reaction based methods are widely used for the identification of the pathogenic bacteria. Yeast and mold, they can be counted on the potato dextrose agar and can cultured on the some fungal media. And pH matters in the growth of the yeast and molds, and it should be lower than the bacterial culture, and so the adjusted pH of the media can be adjusted by adding the 10% of the tartaric acid. For the detection of the virus in the milk, most frequently, PCR is used for the detection of the viruses in the milk. So how the microbial control can be done in the milk? There are various type of the methods which can be used for the control of the microbial microbes in the milk samples. So one of the methods preservation. Usually we do practice refrigeration. Milk can be stored at the refrigeration temperature, which slows down the bacterial growth and quality. Another method is the pasteurization. It reduces the number of the viral pathogens and the spoilage causing microorganisms, and it extends the shelf life of the milk. Pasteurization can be done 60 to 65 degrees centigrade for 30 minutes, or another is at a higher temperature like 71 to 74 for 15 to 40 seconds. Another method is the thermization. It is done by heating the milk at 50 to 7 to 68 degrees centigrade for the 15 to 20 seconds, and rapidly it is cooled down to the 6 degrees centigrade. It is effective against the bacteria which is causing the spoilage, but it does not eliminate all the pathogens. It can reduce the growth of the psychotropic bacteria and can increase the milk's shelf life. Another method is the ultra high temperature. It is used it can kill almost all the bacteria. And by adopting the ultra high temperature, it is used as a sterilization method, and milk can be stored at room temperature for a longer period in the sterile package. In this method, we use generally two types of the temperature, 135 to 140 degrees centigrade for the 6 to 10 seconds for the indirect method, and of 140 to 140 degrees centigrade for 2 to 4 seconds for the direct ultra high temperature procedure. Sometimes preservatives are also used to inhibit the growth or slow down the growth of the microorganism, and these preservatives can be natural by biological origin or the chemical agents. Some of the examples of the preservatives are the potassium dichromate, which can be used with the composition of 0.6%, sodium azide, which can be used with the composition of 0.4%, netamycin. It is a natural preservative, and it is used to the milk for the control of the mold and the yeast growth. Another one is the lactose oxidase. It is a natural preservative which can be used to the milk, and it releases the hydrogen peroxide, and it enhances the natural defensive mechanism of the milk to the control of the microorganism. Another method is the high pressure process. In this components of the bacterial cell, like the cell membrane, ribosome, and the enzymes of the microorganism, they can destroyed by the high pressure and can inactivate the pathogenic and the saprophytic microorganism. It is a non-thermal method, and with the use of the high pressure, bacteria are destroyed in this method. Another method is the microwave heating. In this method, electromagnetic waves are used to generate the heat in the food, and it is very effective in killing almost all the microorganisms present in the milk. Another method is the microfiltration. It can be used in the combination with the pasteurization, and generally in this method it involves the filtration with the membrane with the pore size of the 0.1 to 10 micron, and operates at the pressure of the 0.1 to 8 bars. And it is mainly used to reduce the bacteria, spores, and the somatic cells from the milk, and you extend its the shelf life. Another method is the UV radiation. Electromagnetic radiation with the wavelength of the 10 to 400 nanometer. It is used, and it is used to treat the milk by irradiating it, which effectively kills the bacteria and the other microorganisms present in the milk, and it extends its shelf life. Another method is the milk bacteriofugation. It uses the centrifugal force to remove the microorganism from the milk. It can reduce the number of the bacterial spores in the milk, as well as it can use to the removal of the heat resistant bacteria. And ultrasonic, it is a high power sound wave at the frequencies of the 16 kilohertz and the 100 megahertz. And in this method, sonic wave is passed through the milk, and the changes occur in the pressure, which leads to the cavitation and causes the gas bubble, which is having a bactericidal effect in the milk. Another method is the nitrogen gas flushing technology. It uses the nitrogen gas to extend the shelf life of the milk. In this method, what happens? Normally, the milk contains the oxygen, and when the nitrogen gas is flushed into the milk, it displaces the oxygen, and lack of the oxygen results in the inhibition of the bacterial growth. It is having a bactericidal effect on the certain bacteria and can inhibit the growth of the both type of the mesophilic and the cyclotropic bacteria in the milk. And this method is more effective when it is applied to the fresh raw milk, which is having a low initial bacterial count. Another method is the cold plasma treatment. It utilizes the ionized gas to regenerate the reactive species, which effectively disrupt the cell membrane and inactivate the pathogen. And by inactivation of the pathogen, it sterilizes the milk. It also helpful for the inhibition of the biofilm formation and the repelling the microorganism. So all these methods can be used, and apart from these methods, some another technologies are also coming for the combating the contamination, like thermal biosensor, which is effective for the detection of the on-site microbial detection. Pulse electric field, it is also a non-thermal treatment which disrupt the microbial growth, and it causes the interrupt the microbial reproduction. And another method, some emerging technologies are also coming in the dairy industry, like the patch-based biosensors, and another different types of the eradication techniques are coming. So all these about the contamination. So what preventive measures we can take to control the contamination of the milk? So first of all, important thing, the personal hygiene of the milkers. The milkers which is used to the milking of the cows, they should be of the sound health. Their hands should be clean before the milking of the milk cows. They should wash their hand, and they should obviously should not be with any type of the disease, especially the respiratory disease or the tuberculosis. Animal identification, another method is the animal identification, though, so we should keep constant record of the each animal, whether it is of medical record, feeding record, vaccination record, production record. It should be tagged with the ear tag, so that the complete management of the animal should be done properly, and the proper management of the animals should be done on the farm. Animal housing should be comfortable. It should be with the good natural ventilation, so that when animals are housed together in close, they have a close contact, then infectious contaminants, infectious bacteria, they can pass one animal to another animal and cause the disease to the other animals also. So the animals should be housed in a proper ratio in the stall. It should be comfortable with and cleaning of the house should be done properly. Floors should be properly cleaned, washed, and frequent cleaning of the manure should be done from the houses. Lactating animals should be of the good health. Always consult a veterinarian when if you feel that your animal is having some type of the diseases or infections. Teat, udder, and the adjacent area should be cleaned before the milking, and the pre and the post milking disinfectant should be used for the teat. Milking area, it should be clean, and the equipment should be clean, because the milking equipment are the good, so are a source for the contamination of the milk. Assessment of the milk quality, it regular basis, on the regular basis, assessment of the milk produced in the farm should be assessed for the microbiological quality, and the milk should be stored at the cooling temperature, and so that it can inhibit the growth of the contaminants. And the water, important thing is one another important thing is the water, which is used for the farm for the cleaning or the drinking purpose, or the cleaning of the soil teats, other equipments, and the other flows before and during the milking. After the milking should be very clean, and it should not having any type of the contamination. And the foremost thing is that education and the awareness program should be done for the dairy farmers and the consumers, so that on the personal level they can get aware of the contaminants, and how they can prevent contamination on their own farms. All these can be taught to the farmers. So thank you. All this about the microbial contamination in the milk. Hello.
Yes, ma'am.
It's on my side, it's over, ma'am.
Okay. Okay. So now it's a question session or interaction session. So participants are requested to ask any queries regarding this PPT. So you can text in the inbox also, or can ask directly to the resource person. Someone is there, Doctor? Yes, Dr. Dharmendra Shukla, sir.
Yeah, good morning, ma'am.
Yes, sir.
In your PPT, you have mentioned about the use of cold plasma for
Yes.
removal of bacteria or reducing the number of bacteria. So, is there any practical commercial application available at present? Or it is theoretical
No,
what
sir,
we are learning?
it's a developing technology, and it's as per my knowledge, it on the study basis. It's on the research basis, sir. As far as I know, sir.
Okay. And which particular plasma, I mean, like materials being used for plasma, its matter, or like gases? Nothing. Okay, thank you.
So anybody else? I think there is no questions. So, okay, ma'am, I think there is no questions. And thank you for your nice presentation. I think your presentation was, yeah, it's a very informative session, ma'am. And I think your PPT was so simple that everyone can understand, must have understand it. And that's why there is no questions. So I just thank you for your valuable information, and I am thanking you for your for giving you your so much precious time. So thanks once again, ma'am, and we can now leave for the other work also. So, ma'am, please.
Thank you, ma'am. Thank you for giving me opportunity. Thank you, ma'am.
Yes, sir. Thanks.
Wi-Fi. The cable is not work there.